Your body will manufacture somewhere between 60 and 100 kilograms of ADP today, roughly your own body weight, built and torn apart again before you sleep tonight. At any single moment, you're holding about 50 grams of it, less than a golf ball, and every one of those molecules gets rebuilt once or twice every minute, all day, for your entire life, without a single pause. And the structure doing that rebuilding isn't storing energy in a bond, isn't holding a charge the way a battery holds a charge. It's spinning, mechanically, hundreds of times a second, like a turbine bolted into a membrane inside nearly every cell you have. That correction is going to take some unwinding, because almost everyone, including people who did well in biology class, including some of the people who teach it, carries around a picture of ADP that isn't just simplified, it's structurally wrong. Not wrong the way atoms are mostly empty space, is a simplified but basically fine way of talking about quantum mechanics. Wrong in the sense that the mental model describes a mechanism that does not exist and was never found, despite 2 decades of some of the best chemists of the 20th century looking for it with everything they had, in some of the best funded laboratories on the planet, using techniques good enough to find nearly everything else they went looking for. Here's the belief, stated as fairly as I can state it, because it isn't a stupid belief. It's the belief you were taught, on purpose, by textbooks that use the language anyway. ADP, adenosine triphosphate, gets introduced as the energy currency of the cell, and the currency metaphor gets extended one step further almost immediately. The energy is said to live inside the bond, connecting the last phosphate group to the rest of the molecule. That bond gets called high energy. Break it, hydrolyze it, snap off that phosphate, and the stored energy comes rushing out, like popping the cap on a shaken bottle, and that release is what powers muscle contraction or protein folding, or the neurons firing in your head right now while you listen to this. It's a battery. The bond is where the energy lives. Breaking the bond lets it out. It's intuitive. It's memorable. It's been drawn as a little bolt over a squiggly line in textbook diagrams for the better part of a century, ever since a biochemist named Fritz Lippmann coined that exact shorthand, the squiggle, back in 1941. And it is not how energy release from ADP actually works at the level of chemistry. The strange part is that professional biochemistry figured this out decades ago, and the correction still hasn't fully landed. Once physical chemists pointed out that bond energy and free energy of hydrolysis are 2 different quantities being sloppily merged into one phrase. Most working biochemists quietly agreed the squiggle notation was, at best, misleading. It has mostly disappeared from research papers. It has not disappeared from where you'd actually meet it. Introductory courses, popular explainers, the exact kind of place a curious person first encounters this molecule, because energy hides in the bond, breaking the bond frees it, is such a clean, teachable sentence that it survived its own debunking. That's worth noticing on its own. This isn't a case of the public failing to keep up with settled science. It's a case of settled science failing to evict a metaphor that was comfortable enough to keep the lease. Here's the mechanical problem, and it isn't subtle once you see it. Breaking a chemical bond costs energy. Always, without exception. That's not a biology fact, it's a physics fact. Atoms bond because being bonded is a lower energy, more stable state than being apart. So pulling 2 bonded atoms away from each other requires you to put energy in, not take energy out. If ADP's terminal phosphate bond were genuinely high energy in the way the diagrams imply, a coiled spring, cocked and waiting, breaking it should cost you energy, not hand you energy. The arrow in the diagram is pointing the wrong way. So where does the energy released during ADP hydrolysis actually come from, if not the bond itself? It comes from what happens after the bond breaks. Specifically, from how much more comfortable the resulting pieces are than the original molecule was. ATP's 3 phosphate groups sit crammed together, each one carrying a negative charge, repelling each other at close range, like forcing several magnets face to face against their will. The instant that terminal phosphate breaks free, the repulsion relaxes. The freed ion spreads its charge out through resonance in a way it couldn't while attached. Water molecules rush in to stabilize the separated pieces, releasing energy of their own in the process. And you've gone from one molecule to two, which on its own increases the disorder of the system. And systems tend to drift toward more disorder whether or not any of the energy bookkeeping demanded it. None of that is energy stored in the bond. All of it is energy released by the whole system, settling into a more comfortable configuration once the bond is gone. The same reason a stretched rubber band snaps back, not because energy was stored inside the rubber molecules waiting to escape, but because the system was being held away from its relaxed shape and suddenly isn't anymore. This isn't pedantry, because getting the mechanism wrong at this level gets the entire architecture of the cell wrong right alongside it. If the energy really did sit inside a single bond, a cell would only need one trick: make the bond, keep it intact, break it on demand. A one-step chemical vending machine. That is not remotely what's happening to keep 60 to 100 kilograms of this molecule in continuous circulation. What's actually running in every mitochondrion, in every one of your roughly 30-odd trillion cells, looks a lot less like a vending machine and a lot more like a hydroelectric plant. And once you see the real mechanism, the battery story starts looking almost embarrassingly small next to it. Here's a number worth sitting with, because it's going to reframe everything that follows. Across the inner membrane of your mitochondria, a membrane about 5 nanometers thick, thinner than a soap bubble, there's a voltage difference of roughly 150 millivolts. That sounds unimpressive. A phone charger pushes out more than 30 times that. But divide 150 millivolts by 5 nanometers of membrane, and you get a field strength of around 30 million volts per meter. That's squarely in the range of the electric field inside an actual bolt of lightning. Right now, inside every mitochondrion in your body, there's a field roughly as intense as a lightning strike, held stable, continuously, for your entire life, across a membrane too thin to see without an electron microscope. That field is the real battery, if you insist on keeping the word. Though by the end of this, I don't think you will. It isn't chemical, and it isn't sitting inside one molecule. It's electrical, built into the architecture of a membrane, and ADP synthesis is what happens when that field is allowed to discharge, in a very controlled way, through one specific and genuinely strange piece of molecular machinery. To understand how strange that machinery is, and why almost nobody believed in it for the better part of 2 decades, you have to go back to what professional biochemistry actually assumed through the 1950s and into the 60s. Because the battery-in-a-bond myth wasn't invented for high schoolers. In a more sophisticated form, it was the leading scientific hypothesis held by the people who should have known better. The dominant model of the era was called the chemical coupling hypothesis, and it deserves to be stated fairly, because it was a reasonable guess built on real precedent. Biochemists already knew, from painstaking work on pathways like glycolysis, that cells routinely move energy around using exactly this kind of bond-to-bond handoff. An enzyme breaks one high-energy bond and immediately uses that energy to build a different bond elsewhere, with a short-lived chemical intermediate carrying the energy across in between. It's a real mechanism. It happens constantly in metabolism. So when biochemists turn to oxidative phosphorylation, the process running in your mitochondria right now that uses the oxidation of food to build ADP, they assumed the same logic had to apply. Somewhere between the electron transport chain and the ADP-making machinery, there had to be a molecule, a chemical intermediate, carrying its own high-energy bond, handing that energy directly to ADP the way a baton gets passed in a relay. For roughly 20 years, some of the best biochemistry labs on the planet went looking for that molecule. It should have been findable. Every other intermediate from that era of metabolism, the ones in the citric acid cycle, the ones in glycolysis, eventually got isolated, purified, and put in a bottle on a shelf. Careers were built on the expectation of finding this one. Grant proposals assumed its eventual discovery was a matter of better technique, not a question of whether it existed to begin with. This one never was found. Not because nobody looked hard enough, because it isn't there. It took a specific 15-second experiment to convince the field of that. And it's almost insultingly simple once you hear it. I'll get to it in a minute. Because the person who said the search was pointless in the first place deserves the setup first. And the reaction he got makes the eventual proof land harder. That person was a British biochemist named Peter Mitchell. And in 1961, he didn't propose a minor correction to the chemical coupling model. He proposed that the entire chemical intermediate framework was the wrong category of explanation. That biochemists had spent a decade hunting for a chemical answer to what was actually a physical, architectural question. Mitchell's chemiosmotic hypothesis argued that the electron transport chain doesn't hand off a chemical bond to anything. It pumps protons, hydrogen ions, across the mitochondrial membrane from one side to the other, building up exactly the kind of voltage and TF difference described a minute ago. No chemical intermediate required. The membrane itself, charged like a capacitor, is the energy-carrying structure. ADP synthesis happens later, as a separate step, when protons are allowed to flow back down that gradient through an entirely different piece of machinery. That should have been a tidy, elegant correction. Instead, it triggered what bioenergetics researchers themselves later nicknamed the Ox-Foes Wars. Genuinely one of the most personally acrimonious disputes in 20th century biochemistry, to the point that people reportedly attended conference sessions on oxidative phosphorylation, specifically because they expected a fight to break out and were rarely disappointed. One of the most prominent defenders of the chemical intermediate model, biochemist Edward Slater, spent years arguing directly against Mitchell in print and in person. Mitchell was mocked as hand-waving, accused of dodging real chemistry in favor of vague talk about membranes. A stomach ulcer, worsened by the stress of his academic position, eventually forced him out of the University of Edinburgh entirely. He didn't quietly drop the theory. He used a private inheritance to convert a manor house in Cornwall into an independent laboratory, called it Glynn Research Laboratories, and kept working on chemiosmosis for years with a single close collaborator, Jennifer Moyle, from completely outside the institutions that had rejected him. There was also a quieter disagreement running underneath the loud one, and it's worth flagging now because it comes back later. Mitchell wasn't the only person to notice that gradients across membranes might matter. A chemist named Robert Williams proposed his own version of a proton gradient idea at almost exactly the same time, but with a crucial difference. Williams pictured the protons staying localized, moving along the membrane surface itself rather than spreading out evenly through the surrounding water. Mitchell's version, protons fully mixing into open solution before finding their way back, was the one that became textbook orthodoxy. Williams's version never fully went away. Here's the experiment that changed enough minds to matter, and it started almost by accident, with a result its own author didn't initially trust. André Jagendorf had been studying illuminated chloroplasts, the light-harvesting structures in plant cells, which use the same chemiosmotic machinery mitochondria do, just running in the opposite direction, powered by light instead of food, and kept noticing they produced more ADP than the known chemistry of the electron transport chain seemed to allow for. That's the kind of inconsistency that's easy to wave away as measurement noise, except Jagendorf was careful enough not to wave it away. He took the discrepancy seriously as a real clue rather than an embarrassing error, which is what led him and a colleague, Ernest Uribe, to design a much blunter test in 1966. Strip away the light entirely and see if a phi difference by itself, with no photochemistry running at all, could account for the missing ADP. Their setup was almost insultingly simple. They took chloroplasts and soaked them, in complete darkness, in an acidic buffer at EF4, long enough for the inside of the membranes to fully match that acidity. Then, still in the dark, they transferred those chloroplasts into an alkaline buffer at EF8, one that also contained ADP and inorganic phosphate, the two raw ingredients ADP is built from. No light, no electron transport chain doing any work, no oxidation of any fuel happening anywhere in the beaker. Within 15 seconds, ADP appeared. Sit with what that rules out. If the battery in a bond story were correct, if ADP synthesis genuinely required some chemical handoff from a reaction releasing fresh energy in real time, this experiment should have produced nothing. There was no reaction supplying new chemical energy. There was only a P8 difference, artificially imposed with 2 beakers of buffer, mimicking the gradient light or respiration would normally build. And that alone, with nothing else happening, was enough to drive the synthesis of the molecule every textbook calls the energy currency of life. The membrane didn't check credentials. It just responded to protons flowing back down a slope it had been artificially tilted into, exactly the way Mitchell said it would. This is widely regarded by people who actually work in the field as one of the first genuinely unequivocal pieces of support for the chemiosmotic hypothesis. Not because it was the cleverest experiment ever designed, but because of how little room it leaves for a different interpretation. As Jagendorf's findings started circulating through the field, Mitchell wrote a private letter to Slater, the same Slater who'd spent years arguing against him, essentially conceding that the accumulating evidence was pushing him past hypothesis and toward treating chemiosmosis as an established theory. He didn't sound triumphant. He sounded almost surprised himself at how directly the evidence was breaking his way, writing to the person least inclined to agree with him before he said it anywhere more public. It took until 1978, 17 years after that original mocked paper, for the field to formalize its verdict. Mitchell received the Nobel Prize in Chemistry that year, alone, for his contribution to understanding biological energy transfer through the chemiosmotic theory. Not a shared prize, not a footnote correction to somebody else's model. A solo Nobel for an idea that had spent most of 2 decades being treated as a fringe distraction from real biochemistry. And here's where I have to be honest about something, because presenting this as a fully closed case would be dishonest, and I'd rather you trust the rest of what I tell you than get a tidy ending that isn't quite true. I used to explain chemiosmosis as a completely settled picture. Protons get pumped out, they mix freely into the surrounding water, and eventually, somewhere, anywhere, it doesn't matter where, they find their way to an ATP synthase and flow back through. Fully mixed, fully equilibrated, like dye spreading evenly through a glass of water. That's the textbook version, and for a long time I didn't know there was any argument left to have about it. There is one, and it's the same disagreement I mentioned a minute ago and then moved past too quickly. Robert Williams's localized version, running quietly underneath Mitchell's delocalized one since almost the same year the original hypothesis was published, and it still isn't fully resolved. The question is whether protons pumped out of the electron transport chain actually mix into the bulk water on that side of the membrane before finding their way back. The delocalized picture, the one that won the textbooks, or whether a meaningful fraction of them instead travel along the surface of the membrane itself in something closer to a 2-dimensional slide reaching a nearby ADP synthase without ever fully escaping into open water at all. A review of the accumulated evidence in the mid-1980s by biochemist Stuart Ferguson concluded the honest answer was that the experiments of the time couldn't fully rule either picture out. Newer work hasn't closed the gap so much as sharpened it. Measurements of how fast protons move along membrane surfaces compared to how fast they escape into bulk solution have found surface movement can be substantially faster, and some labs report a detectable TF difference running laterally along a membrane surface, a gradient inside the gradient, that a purely delocalized model doesn't obviously predict. This matters beyond bookkeeping, because if a meaningful share of energy transfer is happening through local, surface-level proton traffic rather than a fully mixed, whole membrane voltage, that changes real numbers. Efficiency calculations, how certain drugs and toxins that target this system actually work, even how these membranes might be organized at a scale below what current imaging can resolve. I'm not telling you this to undercut what you just heard, the core of chemiosmosis. No chemical intermediate, a physical gradient doing the work, a machine that spins in response to it, isn't in dispute. But the fine print, Exactly how protons travel that last few nanometers is still being argued about by specialists more than 60 years after the theory that made all of this legible in the first place. That isn't science failing. That's what science actually being alive looks like, as opposed to the version where every open question quietly resolves itself into a museum plaque. Which brings us to the part of this story I find more remarkable than the gradient itself, because knowing a voltage exists doesn't tell you how a voltage becomes a chemical bond. That's a real engineering problem. Electricity and covalent chemistry are different kinds of things, and something has to physically bridge them. Here's the belief most people land on once they accept that a gradient is involved, and it's a reasonable resting point. Fine, some kind of enzyme uses the gradient to make ADP. In the vague way enzymes are always described as making things happen. That description isn't false, exactly. It's just doing an enormous amount of work by staying vague. The same way saying a car uses gasoline to move is technically true and tells you nothing about pistons. The person who forced the field to get specific was Paul Boyer, working mostly through the 1970s and 80s, using an odd, indirect kind of evidence. He tracked oxygen atoms through the reaction using isotope labeling and watched how ADP synthase's chemistry behaved under different conditions. What he found didn't fit a simple picture of one active site quietly doing its job. The enzyme has 3 catalytic sites, and they weren't behaving independently. They were behaving as though mechanically linked, cycling through different states in a fixed, coordinated sequence. One site loosely binding ADP and phosphate, another clamping down and forcing them together into ADP, a third releasing a finished ADP molecule and resetting. Boyer proposed, years before anyone had a picture of the enzyme's actual shape, that this cycling was being driven by physical rotation, that something inside the enzyme was turning, like a camshaft, forcing each of the 3 sites through the same sequence in turn. He called it the binding change mechanism, and at the time, there's a rotating part inside this enzyme sounded about as testable as there's a ghost in the machine. It stopped sounding speculative in August of 1994, after years spent coaxing a notoriously uncooperative membrane protein into forming crystals orderly enough to diffract X-rays cleanly. The unglamorous, failure-prone kind of work that rarely makes it into the summary version of a discovery. When John Walker's team finally published the structure of the core of ADP synthase, resolved to the level of individual atoms, the structure showed exactly what Boyer had guessed from chemistry alone, years before anyone could see it: an asymmetric, elongated shaft sitting off-center inside a barrel formed by 6 surrounding protein subunits, 3 of them locked into visibly different shapes from one another: open, loose, tight, precisely mirroring the 3 states Boyer's biochemistry had demanded. Boyer and Walker split half of the 1997 Nobel Prize in Chemistry for that combined result. One man predicting a rotor from pure chemical logic, the other photographing it. Photographing a shape that looks like it should rotate is not the same as watching it rotate. In 1997, the same year that Nobel-worthy structure was making headlines, a team in Japan led by Kazuhiko Kaneshita did the part that still gets me every time I think about it too carefully. They took the rotating part of the enzyme, the gamma subunit, and attached a fluorescent filament of actin to it, essentially gluing a visible flag onto an invisibly small rotating shaft, then fixed the surrounding barrel to a glass slide under a microscope. Then they added ADP. The filament turned, continuously, in one direction, more than 100 full revolutions, visible in real time under an ordinary fluorescence microscope, powered by nothing but ADP being consumed 1 molecule at a time. It's a home movie of a molecular machine at work. The smallest rotary motor ever directly observed, filmed spinning like a fan, made of folded protein, a few nanometers across, small enough that thousands of them would fit side by side across the width of a single human hair. Based on how fast the enzyme burns through ADP under normal physiological conditions, biophysicists calculate its actual operating speed, slowed dramatically in that original filmed experiment by drag from the long fluorescent filament used just to make it visible. At somewhere between 200 and 500 full rotations every second, tens of thousands of rotations per minute, from a machine you could never see without special equipment spinning inside you right now. That is the actual device converting a voltage into a chemical bond. Not a vague enzymatic use of energy, but a physical turbine, protons flowing through it the way water flows through a hydroelectric dam, torque generated by that flow forcing 3 catalytic sites through a mechanical cycle that squeezes ADP and phosphate together hard enough, and in exactly the right geometry, to form a new covalent bond. Measured directly under load, that rotor produces something on the order of 40 piconewton nanometers of torque with each step, a unit so far outside daily experience it's almost meaningless on its own, except for what it implies. For an object a few nanometers across, that's enough twisting force to do the job with room to spare, which is part of why the same enzyme, run in reverse, can also burn ADP to pump protons the other way, something bacteria rely on when they need to power a flagellum instead of build a phosphate bond. It's a motor that can be driven from either end, not a one-way tap. The bond isn't releasing stored energy when it eventually breaks. It was physically built, under mechanical force, against its own preference not to exist. The same way you'd press 2 magnets together, against their natural repulsion, which is close to literally what's happening at the catalytic site, given how much those phosphate groups don't want to be near each other in the first place. You'd be forgiven, at this point, for assuming the story is basically finished. Filmed, crystallized, Nobel recognized twice over, reduced to a rotation speed you can quote in revolutions per second. Structures this thoroughly characterized don't usually stay interesting, and yet this exact machine currently sits at the center of one of the more unsettled arguments in cell biology, one that's been actively contradicted and recontradicted in the scientific literature within roughly the last decade, under certain pathological conditions. Calcium overload, the kind of cellular stress that happens during a heart attack or a stroke, when tissue is starved of oxygen and then suddenly reperfused. Mitochondria can undergo something called the permeability transition. Their inner membrane, the same membrane holding that lightning-strength voltage, suddenly and catastrophically becomes leaky, flooding open to molecules that should never cross it, collapsing the gradient entirely and helping trigger the death of the cell. For years, nobody knew what physical structure was actually forming that fatal pore. The leading suspect, based on a growing body of evidence over roughly the last 15 years, is ADP synthase itself, the same rotor that spends its normal life building the molecule that keeps you alive, apparently capable, under the wrong conditions, of reconfiguring into something that helps kill the cell it's inside of. Exactly how that reconfiguration happens is still being fought over, in real time, across the primary literature. One research group, led by Paolo Bernardi, has argued the pore forms at the interface between paired ADP synthase